welcome to the third lecture of week 3 of MOOC's course on Advanced Analytical Technique. In the second lecture of the third week, we have discussed about the different components of inductively coupled plasma. We have witnessed different types of nebulizers. We have also discussed about a new type of nebulizer called as the Frit nebulizers, which utilizes a glass membrane, and those Frit nebulizers have a counter current flow, means gas is coming from the top while the liquid sample is coming from the bottom. Also, we have briefly discussed about the pneumatic nebulizers as well as ultrasonic nebulizers. In pneumatic nebulizers, we have one of them is called as the concentric nebulizer, where in a single tube we have aerosol, where in a single tube we have sample as well as argon gas, and they are mixed and then they are entered into the spray chamber. While in case of the cross flow, we have one tube which allows sample to pass through it, while at 90° we have another tube in a cross flow pattern which allows argon gas, and both are being mixed into the spray chamber to convert the aerosol. Then after this nebulizer, we have discussed about the plasma source, the ICP torch, and then we moved on to monochromators. Now we'll discuss about the two common types of spectrometers that are being used in atomic emission spectroscopy. These two spectrometers are called as sequential spectrometers and simultaneous spectrometers. From their name, we can easily perceive that sequential spectrometers, they basically works upon the principle of sequence, means these spectrometers are used to, first of all, we allow one light radiation to pass through it, the light which is coming from the excitation of the sample, we allow one particular wavelength to pass through it and finally falls on the detector. Then we change the sample, then we again go for another light radiation to identify another element. So this means that we can identify different element. For example, if we have multiple elements present in a system or in a matrix, we have to do the analysis repeatedly, then only we can be able to identify or determine all these elements. Of course, it is a tedious task which requires long times, although the machine is inexpensive because all basic equipments are used, like we have one single photomultiplier tube as a detector, we have one monochromator. Similarly, since we are not doing the multiple analysis at a time, we may not need a very high excitation source, and therefore although it requires a skilled operator, but very highly trained person is not needed for these sequential spectrometers. On the other hand, we have simultaneous spectrometers. These spectrometers have the beauty that we can go and perform analysis of multiple elements at a time. We can detect multiple elements simultaneously. Therefore, these simultaneous spectrometers are very quick in response. We can, just by doing one or two run, we can identify numerous samples. We can go up to 30, 40 or 50 elements in a sample by these simultaneous spectrometer just in few minutes. But of course, since they require multiple element analysis, so they use various type of monochromator, or I should say, instead of a monochromator, they have a polychromator which allows different type of light radiation to pass through it. And then of course they have multiple photomultiplier tubes or some other sort of detector that can detect all these elements at a time. I'm not going to tell you the name of that detector which is being used. That we'll discuss during this presentation in the later slides of this particular topic. As I said, sequential spectrometers, they are basically used for single elemental analysis at a time, and for the second element we have to perform the analysis again. So we have movable grating to select relevant wavelength. We move the grating, select particular wavelength with the help of the entrance and exit slit, and since they are used to detect single element at a time, they uses a single photomultiplier tube to detect. For the second element, We again draw put the sample which is being drawn with the help of peristaltic pump into the nebulizer, and it again gets converted into aerosol, which is then injected into the spray chamber and finally reaches to the excitation source. Upon excitation it emits light, and the light radiation that is being emitted, again it passes through monochromator. We allow specific wavelength to pass through it, which falls on the detector so that the second element can be detected. In this way we do the analysis. Sometimes these monochromator uses normal grating, but sometimes we prefer holographic grating, which has large number of grooves, which can narrow down the light radiation to a greater extent. So these spectrometers are designed to take measurements turn-wise or in sequential order, means one by one. First analysis is done, then we go for the second elemental analysis. However, due to their time-taking protocols, when these sequential spectrometers were developed, there were high demand from the industries as well as from research institution to reduce the time and to enhance the possibility of detection of multiple element. So just after the development of these sequential spectrometers, there has been some new developments in which a slew scan spectrometers have been developed. These slew scan spectrometers are an advanced version of these sequential spectrometers, where we have a little bit quicker analysis time, which ultimately shortens the whole protocol. And now with the development of this slew scan, we can perform analysis of 4, 5 or 6 element in a short span of time. In this particular figure, I am showing you a sequential spectrometer, but there is no point of showing the nebulizer and all those components like the nebulizer, peristaltic pump or spray chamber, because our point of discussion here is when the light radiation has been created, and in case of ICP or in case of AES, the light radiation is created after the excitation source. So this is our excitation source. When the sample is excited, light radiations now are coming. They passes through a prism, and then again we uses mirrors to direct it towards these gratings. These are holographic gratings. We have a normal grating like this. In holographic grating we have some thickness as well. We have this type of plates which have some thickness, and then on these plates we have these grooves created. When these light radiation falls here, they are distributed into different wavelengths. Then they again passes through a refracting mirror, and finally they passes through the exit slit, and they are detected on a detector. Sometimes we use dual detector for even more reliable analysis. For the second sample, for the second element, we again carry out the same procedure. The sample is being injected into the atomizing source from the spray chamber, and then in this atomization and excitation source, means the plasma torch, the sample is excited. Again we have some light radiation. This time we move the grating as well as entrance slit to select some other wavelength corresponding to another element, and finally when that radiation passes through the exit slit and falls upon the detector, we again we are able to analyze or determine the second element. So in this way we can perform repeated analysis for multiple elements. Now in order to complete this sequential analysis, the monochromator grating or even the holographic grating is rotated in a controlled manner. We rotate it in a controlled manner and select different wavelength, so that next time we have a different element which is being detected. Then third time we rotate it and allow the third wavelength to pass through it and falls on the detector to detect the third element. In this way we have to keep moving the monochromator grating so as to select different wavelengths and light radiations. That ultimately helps us to identify different elements. And then in order to reduce the tedious protocol, as I said, slew scan spectrometers have been developed, which allows very easy and controlled movement of these monochromator so that we can perform the analysis even in a short period of time. These slew scan spectrometers, they reduces the longer analysis time of sequential spectrometers. So we can reduce the longer analysis time of sequential spectrometers by using these slew scan spectrometers. These slew scan spectrometers have slew scan detectors that have a combination of monochromator and slit to select a specific wavelength. We already have a combination of monochromator and slit. In the first case, we select one combination of monochromator slit which allows a particular light radiation. Then we move the knob, just like we have a knob over there, just like we have our volume knob in our uh, systems. So we move that knob, we select a different combination of monochromator and slit, and this way we change, and in this way we keeps on changing the combinations of this monochromator and slit, and we can very easily go to the next combination, thereby selecting different wavelengths that ultimately helps us to analyze more samples in a short period of time. So because in the previous case, in this one we have to every time change the position of these slits and in order to select a different wavelength. But in case of slew scan, which a combination is already fixed, by just by rotating the knob we can select different combination of slit and monochromator, means different combination of grating and slit that allows a different light radiation to pass through it, and a new element has been detected. Then again we rotate the knob to adjust or to fix another combination of slit and grating, and then the third radiation is selected while the first two are being excluded. When this third radiation falls on the detector, the photomultiplier tube, the third element is detected. Although in this case also we have to rotate the knob, but rotating this knob is very easy to select different combinations, and therefore the time for analysis of multiple elements is quite reduced as compared to the sequential spectrophotometers. Then we have the simultaneous spectrometers. So in case of sequential spectrometers, we have a normal sequential spectrometers, and then we have a slew scan spectrophotometers. Although in both the cases we have to set different combinations, but in case of slew scan, the selection of new combinations of slit and grating is quite easy. Just by rotating the knob we can select different combinations very easily and very quickly as well, so that we can identify element after element. We can identify different element one after the other in a short period of time. While in case of the old version of these spectrometers, which are called as the sequential spectrometers, we have to every time adjust the grating as well as every time adjust the, so as to select different wavelengths and different light radiations in order to determine or detect all the elements present in the sample. Now this slide shows the simultaneous spectrometers that are basically known for simultaneous analysis of multiple elements at a time. Now, since we are doing simultaneous analysis of multiple elements, means for example, if we have a sample from river water which is close to an industry, and there are some news that that industry is polluting that river, and we have to analyze the sample to identify all the heavy metals present in that particular sample which is coming from a particular river. Or for example, if we have a blood sample of a person who is having some type of poisoning, so in that case as well. Further, in case of drinks like juices or soft drinks, in order to identify the possibility of different elements, we may use these simultaneous spectrometers. And since we are going for the detection of multiple element, that too at a particular point, we need number of monochromators, or I should use the word polychromators, and we also need multiple detectors. So there are two possibilities: either we can use new either we can use multiple PMTs, means the multiple photo multiplier tubes, which you know that they are photosensitive. They have a photocathode and photoanode placed on the either side. Photocathode once captures the photons that are coming from into the that are present into the light radiation, they convert it into an electrical signal, means electrons are ejected by photoelectric effect. That photocathode is light sensitive, and those electrons, of course, they are not very accelerated, and the number of electrons are also low, so they first of all falls on the dinodes, which creates secondary electrons, and these accelerated secondary electrons, they falls on the cathode, creating current, and a peak is obtained. So this is how a PMT works. Just I wanted to do a recap to make you realize what PMT is. The other type of detectors that are even more useful and even more authentic and reliable are called as the solid state array detectors. I told you in the beginning of this lecture that I am not going to tell you the name. I'll tell you the name once that particular slide comes in. So now the second type of detector that are used for multiple analysis are called as the solid state array detectors. These solid state array detectors, they basically work on charge coupled devices. Charge coupled devices. There are multiple types of solid state detectors. We may have solid state detectors that works on charge coupled devices, CCD, and we also have solid state array detectors that works on charge injection devices. But and both these detectors can be used for the analysis of 50 or 60 elements at a time. In their spectrum, we can have hundreds of spectral lines, and these detectors can be able to identify so much lines, because one element can have more than one spectral lines present in the sample, because sometimes the excitation occurs from E0 to E1, sometimes it happens to E2, while in some other case it happens to E3. So when in AES the electrons are coming back from E3 to E0, the light radiation emitted, of course, are having more energy or shorter wavelengths. Then when from E2 it is coming, they have some different wavelength, and when from E1 they are coming, they have some different wavelength. It is up to us that we can deselect or exclude any two of these wavelengths. But if we want, we can have all these wavelengths in the form of spectral lines in the spectrum. So therefore I am saying that with charge coupled devices or charge injection devices based solid state array detectors, we can go up to 50-60 elements detection at a time, and you can now calculate that if for one sample we can have 3 or 4 lines, then how many lines may be present in the spectrum. Of course, that creates some complications, but still they have the capability to go up to so many spectral lines, and those can be detected as well. So as of now, the most advanced atomic emission spectrometers that are based on inductively coupled plasma or other type of plasma like the direct current or the microwave plasma, they used these solid state array detectors. But of course, since these SSAs are very sensitive devices, they are highly costly. So the instrument that have these SSA for multi-element analysis simultaneously, of course, they are very expensive. They are very expensive. Now let us move on to discuss about the polychrometers. So there are two types of simultaneous spectrometers from this discussion: those which utilizes polychrometers and those which utilizes solid state array detectors. Now what are polychrometers? Polychrometers have a fixed diffraction grating, means they have a diffraction grating that is being fixed, but they have multiple exit slit. For example, we have a light radiation of wide wavelength is coming. We have an exit slit that allows this light radiation. Now we have a mirror over here, and then when this mirror reflects this radiation backward, we have a grating placed over here. sometimes we use eschel grating as well. This is a particular type of grating which is used for dispersing light radiation into individual very narrow wavelengths. So when this light ray falls on the grating, it is converted into different light radiations. Now in case of monochromator, we have only one exit slit, but now in this particular case we have multiple exit slit that allows more than one light radiation to pass through it. Of course, it does not mean that we cannot stop any light radiation. We can identify some, we can exclude some light radiations, but still we can allow at a time 4, 5 or even more number of light radiations to pass through it with the help of these multiple exit slit. They are multiple exit slit. Now the question is how these multiple light radiations that are passing through these multiple exit slits are going to be detected, and the answer is multiple PMTs installed in this particular case of spectrometers, in this particular case of simultaneous spectrometers. So when these multiple lines are passing through, so when these light radiations are passing through multiple slits, we have multiple detectors placed over here in front of these in front of this polychromator, so that they can detect each photomultiplier tube detects a particular wavelength confirming the presence of a given element in the sample, and we can go up to 60 elements. Can you believe this? 60 elements can be detected at a time with the help of these polychromators based simultaneous spectrometers. So this is a design of an ICP which is based on a polychromator. We have a plasma source, as I said earlier, since we are focusing on monochromators and polychromators which are used for the selection of particular light radiation or selection of multiple light radiations. There is no point of showing nebulizer as well as the spray chamber in this particular diagram. So this is the plasma torch, which is also shown in very small size, just to give more space for these component like the polychromators. Then the light radiation that are coming, they are passed through an aperture, and then finally we have mirror that reflect this light radiation. Then we have concave diffraction grating, or sometime we have a shell grating. Now in case of this polychromator, we have multiple exit slits. These multiple exit slits, they are placed on a circle. This circle is called as Rowland circle. This circle can be moved very easily, and by moving the circle we can put different slits in front of this light radiation, so that we can pass multiple light radiations in a single run. And once this light radiation passes through the exit slit, we have different types of PMT devices. The photomultiplier tube, the position of the photomultiplier tubes can be moved up and down, left and right, so as to capture a particular wavelength, and in this way the analysis can be done. These photomultiplier tube are connected to our output device. They capture photons, they capture photons, converting them into an electrical signal, which is then amplified and finally presented in the form of peaks on the spectrum. So this shows how a polychromator works. So a polychromator works by taking a Rowland circle on which we have multiple exit slits. We have a grating, either the concave diffraction grating or eschel grating. This grating converts light radiation into individual components, and then with the help of this Rowland with the help of this Rowland circle, we move these multiple exit slits in order to select different wavelengths. And finally we have these photomultiplier tubes which can be moved up and down. We have multiple photomultiplier tubes here, and these photomultiplier tubes then capture those radiations coming from the different multiple exit slits and convert them into electrical signals. So in this way a polychromator works. So basically a polychromator means we are having multiple monochromators, and this works when we have a grating and multiple exit slits. We don't need multiple entrance slit, and only multiple exit slits are needed, which allows multiple light radiations to pass through it. And in order to have a combination that can easily be, and in order to have a combination that is easily movable, that can be easily moved to select different wavelength, we have both these devices placed on the Rowland circle. This is again another type of a polychromatic device where we have a polychromator used, but in this case we do not use a concave diffraction grating like we have in this particular case, but in this case we use an axial grating. This is an axial grating where we have, this is in the, like this, we have this axial grating like this, which, so this is our axial grating, which is like this. We have grooves on this plate which convert light radiation into individual component, but there is a peculiarity in this particular design. I hope you have identified in this diagram that we are using a grating as well as a prism. Now when we are using both these uh, in combination, means we are using a grating as well as a prism, it means we are further narrowing down the wavelengths of the light radiation. For example, if we have a light radiation that is covering different wavelengths ranging from 300 to 400, say for example, so when it enters, first of all the light ray that is coming, it is reflected with the help of the source mirror that allows it to enter through the entrance slit, and then we have a culminating mirror which again reflect this light radiation to the axial grating. Now when it falls in the axial grating convert this light radiation into individual radiations. For example, we have from 100, it converts its different 100 wavelengths to different individual light radiations. Then we have a prism that further reduces or narrow down the wavelengths of these radiation, and then we have an aperture plate placed over here which has different holes. Now through these holes different light radiation passes through the sample and falls on the multiple PMT devices, which detects these multiple devices, which detects multiple elements simultaneously. So here in this case we can go up easily up to 60 elements. So in case of this particular combination, we have to move this Rowland circle or rotate this Rowland circle to select different wavelength coming from the multiple exit slit, so that multiple elements can be detected. But in this particular case, when we are using an axial grating and a prism which narrow down the light radiation to even a smaller interval or to even a smaller level, and then it passes, it is, they are being reflected from the plane mirror, and finally there is a plate aperture where we have different holes which allows multiple light radiations to pass through it simultaneously, and then we have multiple PMT devices. So here we do not need to move anything or move a device or a circle like a Rowland circle. We can very easily make these multiple light radiations to focus on the PMT device. Of course, that are also multiple PMT devices, so we can very easily have these light radiation focusing on the PMT devices, and multiple elements can be detected in a single run simultaneously. So we can say that polychromators makes our work very faster, and the data is also, and the result is also quite reliable. However, the cost goes up because we are using polychromators which have multiple slits. Sometimes we are using axial grating which has a grating and a prism together, and also the most important thing, since we are using polychromator, we need multiple detectors, which makes the instrument very expensive. So if we are having a budget limitation or a small industry which cannot go for this particular equipment, so they may satisfy themselves by using a sleeve scan spectrophotometer or just a sequential or just a sequential spectrometer that can be used for having single analysis at a time. But for a multiple analysis system, we need these polychromators or the sequential spectrometers, where we have polychromators that are placed on a Rowland circle, or those polychromators that uses eschel grating and a prism, which narrow down the light to a greater extent, that passes through a plate aperture and finally detected based on different types of and finally detected based on multiple PMTs, so that at a time we can have multiple element analysis. Although this, since we are going for large number of equipments, since we are going although but since we are going for large number of samples at a time, so we may have more spectral lines which may complicate the spectrum. Then there comes the last topic of today's discussion, the solid state array detectors. In case of these solid state array detectors, as I said, there are two types of solid state array detectors, one which uses charge coupled devices and the other which uses charge injection devices. We mostly prefer solid state array detectors that uses charge coupled devices. Now these charge coupled devices are basically light sensitive materials, or you can say they are sensors that works on the principle of photoelectric effect. For example, a small one single light sensitive instrument is like this. When the light falls on it, it basically captures light photons and they work on the concept of photoelectric effect. When the light photons falls upon these light sensitive plates which are connected to capacitors, electrons are created, means electrons are ejected. As you all know, that photoelectric effect means when light photons falls on a metal surface, electrons are ejected. When electrons are ejected, they leave behind holes, so electron hole pairs are created. These electron holes pairs create an electrical signal which is then stored into the capacitor which is connected with these devices. Therefore they are called as charge coupled devices, because these charge pairs are created, they are being stored in the capacitor, and an electric current or electric signal is generated. And this is only one device. There are multiple devices that are connected to form a array. Array means a series of those sensitive elements connected with each other, that can be used for each single element is used for a single wavelength, means each this sensitive element can be used for one elemental analysis. So they are highly sensitive, they have very fast response time, excellent wavelength resolution, means with this we can have different wavelengths that are very easily resolved, very clearly resolved from each other. And therefore these solid state array detectors are useful for instrument like ICP OES, inductively coupled plasma optical emission spectroscopy. They are also used sometimes in UV visible spectrometers and in Raman systems. These machines, these solid state array detectors are also useful in and these solid state array detectors are also useful in medical imaging. They are also used to study about the different types of cells, and they are also useful in remote sensing. So they have multiple applications in different areas. So a solid state array detectors, they are always used with polychromators, means either they are connected to Rowland circle which have combination of multiple exit slits and concave diffraction grating, or they are used with eschel grating, where we have eschel grating and a prism that narrow downs the light to a greater extent, and then we have a plate aperture which allows multiple light radiation to pass through the small holes, to pass through those small apertures of that plate, and then those multiple light radiations then falls on the detector. So these solid state array detectors are always used with polychromators, and therefore they are part of simultaneous spectrometers. So this is a type of solid state array detectors where we have different light sensitive elements placed, or light sensor. This light sensor is shown here. These light sensitive elements, sometimes called as pixel or light sensitive elements, when they receive light photons h nu, they create electrons that are being ejected, and then we have a holes remaining. This electron hole pair create charge carriers, and these charge carriers are connected with, because in between these we have capacitors that stores this charge, and ultimately the charge is released and an electrical signal is generated, and that electrical signal is then amplified, and finally a signal or a peak is obtained in the computer or the readout device. Here you can see we have a plasma torch. The sample is excited, the excited light radiations are coming, and they are first of all focused with the help of these mirrors to pass through the entrance slit, and then we have, so then there is a parabolic culminator which focus this light radiation to the HL grating, which is having these types of grooves which converts this light radiation into smaller light radiation, into light radiations of smaller wavelength, or I should say into individual wavelengths. Then they again passed through a visible prism, and then finally they focused upon a detector which is based on solid state detector, and it has the capability to detect multiple elements at a time. So this is the flow sheet of a solid state array based detector, which uses a polychromator either in the form of a Rowland circle, which has a concave diffraction grating and multiple exit slit, or it has an HL grating with a prism in combination and a plate aperture that allows multiple light radiation to pass through it. Either of these two polychromators are used, and they are coupled with solid state array detectors so that we can detect multiple elements at a time. So with this, we have discussed the different components of ICP, inductively coupled plasma, right from the beginning, that is the peristaltic pump, the nebulizer, spray chamber, its torch for creating the inductively coupled plasma, then the monochromator, the type of spectrometers that uses monochromators like our sequential spectrometers, and the slew scan spectrometers slightly reduces the time of analysis. And then we have sequential spectrometers that works on polychromator or the polychromatic devices that you either use multiple PMTs, multiple photomultiplier tubes for detection, or they used solid state array detectors, which is a combination of light sensitive material based on charge coupled devices, CCD. So with this we have completed the instrumentation of inductively coupled plasma, AES, where we there and that probably be the last lecture of this week, where we'll discuss about the different types of spectral and non-spectral interferences possible in case of ICP. Till then, take care. Thank you very much.